All right, students, very good morning. So today we're going to have a PYQ session, that is a previous year question solving session with respect to the chapter biotechnology and its application. So yesterday I told you that this chapter is small but very effective, and you're going to get the questions that you're already aware of, but they might lead to a small confusions where you may mistake the answers. Okay, so let's be competitive and have this nice sportive spirit, and let's move on with our session today. So each question you already know that we have 1 minute to solve these questions. So I would recommend you be fast, read, understand, and try to answer. Okay, and it would be much appreciated. So here we go. Here we have our first question. Which one of the following techniques does not serve the purpose of early diagnostics of a disease for its early treatment? So this was 2023 question. Recombinant DNA technology, serum and urine analysis, polymerase chain reaction, that is PCR, enzyme linked immunosorbent assay, or ELISA technique. Okay, so just bracket this ELISA technique. So you have 1 minute to answer this question. So let's go, students. Let's see what you got. Everyone should poll. Who will answer this question? Which one of the following techniques does not serve the purpose of early diagnostics of diseases for its early treatment? So you have the options and you can answer. Either you can answer me in the chat box, even that is okay. What's the answer you're saying? Nothing? To put it in the chat box. That's better. Chalo. So the answer for this question, okay? So you try to answer this as early as possible. If not, it's fine, okay? So see, the question asks which of the following techniques does not serve the purpose of early diagnostics? See, early diagnostics means you have to get the results as accurately and as fast as possible. So the among the given options, you can see that option B, that is serum and urine analysis, will take a lot of time. Why it takes a lot of time? The reason is if you take a blood serum analysis or if you take urine analysis, okay, when you do the tests, basically what happens is you cannot be sure what is the type of an organism. So again, they will incubate it, okay, culture these microbes, detect the strains, and then come to a conclusion this is a causative agent. So it's a very long procedure. It might take weeks for us to get the results. Whereas if you talk about rDNA technology, or if you talk about PCR or enzyme linked sorbate assay, so in all these things, so our recombinant DNA technology we are using for molecular analysis. Next one, PCR, you know that amplification, through amplification we can detect. Enzyme linked, immunosorbate assay is a immunological assay. Even in this case, it will lead to rapid tests or rapid analysis of a particular disease. Okay, so our option is B. It does not serve. Serum and urine analysis is very, very late. That's why these techniques were developed. Okay, next one. Transposons are, okay, wait. Transposons can be used during which one of the following? Gene silencing, autoradiography, gene sequencing, and PCR. So here are your options. So the question is simple. Transposons can be used during which one of the following? So here is your options. So let's see what you have. You have an answer? Come on, chat. We have students here. Guys, what is the answer? Want the question? Here you go. I'll flash the question for another 10 seconds. Transposons can be used during which one of the following? Gene silencing, autoradiography, gene sequencing, and polymerase chain reaction. Okay, now you know the options. Okay. C. Good. Finally, Veera Venkataraju, you're awake. Yeah? Good. So let's see if you're right. Gene sequencing. No. Actually, what are transposons? So transposons are jumping genes. Okay. So basically, what happens? Maize, Jannamukka, maize. So if you see that, you know that it is yellow color kernels will be there, right? The seeds, what we eat, it's like yellow color. So there are certain situations where you can end up seeing this Janamuka or corn or maize coming in different colors: purple, red, pink, orange, blue, green. All these different colors will come in the same kernel. Now, when you look at that, you will be amazed, like, how is it even possible? It can't be so many colors. Either it should be one or two colors, but you're seeing 20 different colors. How is it possible? See, what happens is, if you have a gene, okay, let's say this is a different genes, okay. Now, transposons are jumping genes. So they have elements called as transposable elements. What they do is they jump from one gene sequence to another gene sequence, another gene sequence to another gene sequence, and they keep on expressing them. Get my point? So similarly, why we use transposons or jumping genes? So we know that when an organism like Melodigyne incognitia infects our tobacco plant, what it does is it has an infection cycle where it produces pathogenic RNA. Okay. So now this RNA should not be allowed because it will lead to the pathogen proteins, right? So that will help this pathogen to proliferate within the tobacco plant. So what are we doing? We are using transposons, and transposons will see there is a long sequences of DNA genes. There are so many genes here and there. What it will do? Oh, sorry, where is it? Yeah, here and there, what it will do is, for example, see, it will go and read this sequence. It will go read this sequence. It will go read this sequence. So ultimately, what will happen? Let us say that the pathogenic RNA is having a sequence of UUCCGAUC. Okay, this is the sequence of RNA of pathogen. Now, as it jumps from one element to another element of a gene, it will see what is the complement of this. It doesn't know, but ultimately it will be producing a complement of this. So finally, our gene sequence should be, for you, it should be A, and then G, C, U, T, and then A, and then G. So when it comes across this sequence, now what will happen, people? So it will form a complementary RNA for against this sequence. So when it forms a complementary sequence against this sequence, now this and this, like for example, the same RNA about this will make a double stranded RNA, thereby silencing the gene. So this is called as gene silencing technique, right? So gene silencing is also called as RNA interference technology or RNAi technology. So your option should be A, okay, not C. Okay. Do you have any other suggestions or answers? One second. Nothing. Okay, let's go for the next question. And our next question is, see, all of these are previous year question, students. So you can see it's asked in 2022. So let's see what the question is. In gene therapy of adenosine deaminase deficiency, okay, the patient requires a periodic infusion of genetically engineered lymphocytes because, okay, so our options are here. Gene isolated from marrow cells producing ADA is introduced into the cells at embryonic stages. Lymphocytes from the patient's blood are grown in culture outside the body. Genetically engineered lymphocytes are not immortal cells. So retroviral vectors is introduced into these lymphocytes. So here is a question. Just read it carefully once again, and I'm going to help you understand this. Okay. So the question asks, in gene therapy of ADA or adenosine deaminase deficiency, the patient requires a periodic infusion of genetically modified lymphocytes. Why? Because. So your options are gene isolated from the marrow cells producing ADA is introduced into the cells at embryonic stage. Lymphocytes from the patient's blood are grown in culture outside the body. Genetically engineered lymphocytes are not immortal cells. Retroviral vectors is introduced into these lymphocytes. So I hope this is enough to understand the question and to understand what are the options. Okay, let's see if you have got it. Okay, I will leave the screen exposed for another 10 seconds. Read the question carefully. Try to learn the answer. Okay, exactly in 30 seconds, I'm going to show you what is right. Okay. Okay, did you get the question, students? Can we go for the answer? Yes. Do you have an answer? No. Okay, no problem. Let's see. Okay, now what happens in gene therapy of ADA deficiency? Now, what we do is we take the lymphocytes. So we take the lymphocytes, which are the stem cells, and then we genetically engineer them by inserting a healthy ADA gene through our retroviral vectors. So once we have a recombinant lymphocyte cells, RLCs, okay. Now what will happen? We will clone them and we will increase their numbers. Correct, right? So once we have sufficient numbers, now these cells can be ready for the infusion. So infusion means you can just put it into the patient's body. Now what happens is the lymphocytes that we have extracted from the patient, naturally these lymphocytes are not immortal. Basically they die after a certain period of time. So again a new set of lymphocytes have to be injected into these patients. So that is why what we will do, we will bang this. I told you about this, right? So once we have created the recombinant lymphocyte cells, what we do is we clone them into a large extent and we keep the reserve of them. So after a certain period of time, regularly this patient can go get the lymphocyte infusion done and come back and lead a healthy life. Okay, and the reason is simple. Whenever these cells are introduced, genetically engineered cells are introduced into our body, they die after a certain time. Even naturally, also lymphocytes cannot live for longer period of time, or it will not live forever until you are there. So it will only have a certain period of lifespan. So after that lifespan is done, you need to induce a new set of lymphocyte cells. So that is why, basically, why we have to see why the periodic infusion. Infusion means like you just put it into the patient's body. Okay, infusion like blood infusion. How do I tell? Same way. So infusion of genetically engineered lymphocytes. Because why do we have to do that? Because they are not immortal. Genetically engineered lymphocytes are not immortal cells. So that is why we are doing this technique. Okay, understood? If not, you can ask me. Okay. So here is your next question. Our next question is, which of the following is not an application of PCR, that is polymerase chain reaction? And your options are purification of isolated protein, detection of gene mutation, molecular diagnostics, and gene amplification. Okay. So let's see. Which one of the following is not an application of PCR? Okay. I hope this is helping you kids, okay? In spite of you not answering, I hope this is helping you. But yet I'm leaving some space and time for you to answer so that it'll be nice for you to participate. So, but it's okay if you don't answer, but if it's good if you're listening and if you're understanding. Okay. So let's see what is this question about. So it is not an application of PCR, polymerase chain reaction. Again, it was asked in 2021. So if you see the last three of the options, that is gene amplification. Do we do that? Yes, that is what is polymerase chain reaction. Molecular diagnostics. Do we do molecular diagnostics? Yes, we do molecular diagnostics. Next one is detection of detection of gene mutation. Yes, we do detection of gene mutation. So even though we do SSS for all these three things, I'm going to justify how it is done. But when it comes to purification of isolated protein, we do not use PCR. Okay, so we don't do use pure PCR. We use downstream processing. Anyways, coming to our polymerase chain reaction, coming to the detection of gene mutation. See, what happens is whenever we have a healthy gene, so let us say that we have a human being, and human being A is healthy and human being B is unhealthy. I'll write it as UH and I'll write it as health, H, okay? Now let's say that we have a gene, and you can take any genes, okay, of any hormone or an enzyme or a protein, etc. So the most common thing, or let's say it's a growth hormone, okay? So for production of growth hormone, okay, it's an example, okay, kids, okay? So this for a production of growth hormone, what do you need? You need a gene. So let's say that we have a gene, growth hormone gene, correct? Okay. Now let us say that B has a mutated growth hormone gene, and let's say that this person is having gigantism or dwarfism. So what happens? The gene sequence should be altered or mutated for him to have such a disorder. Correct, right? Overexpression or underexpression will happen. So let's say that this part of his gene is mutated. How do we detect them? So when we take these genes, okay, when we take this gene and we amplify the genes, when we amplify means create millions of copies of it or thousands of copies of it. Now what will happen? The short fragment of this DNA, what has happened to it? It has amplified or it has increased in its volume. Now, when we cut it and undergo electrophoresis, or if we go for radioactive hybridization, so how do we do it? We take our healthy gene, we label it with radioactive labeling. Okay, we take radioactive labels and we label them. So this will get radioactively labeled. Now, this is called as a probe. And we take this probe and we hybridize it. When we hybridize it, all the sequences that are not mutated will be hybridized. And the sequences which is mutated will not be hybridized. So the short fragment of sequences which remains unhybridized are called as mutated sequence. Now that will help in gene detection of gene mutation or molecular diagnostics of a disease. Okay, so these two. And we know what is gene amplification. So PCR stands for polymerase chain reaction, which involves three essential steps, that is denaturation, annealing, and then it has extension. After extension, we have amplification, where with a small piece of a DNA, okay, so even with a small fragment of a DNA, what we can do is we can create millions of copies of DNA, okay, within a very short period of time. So amplify means multiply or increase its number. So that is what we are doing with respect to our gene amplification. Okay, I hope it's clear. Yes. So give me a reaction, kids. Like, are you understanding? Are you able to listen to my voice? Is my voice audible? Yes. I want a response from you. Good. 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 And this is enough. Okay. So it's helping, I feel. Let's go next question. Okay. Here is our next question. When gene targeting involves gene amplification is attempted in an individual's tissue to treat a disease, it is known as molecular diagnostics, safety testing, biopiracy, and gene therapy. This is a very, very easy question. So the question is very simple. When gene targeting involves gene amplification in attempt to is attempted in an individual's tissue to treat a disease, it is known as molecular diagnostics, safety testing, biopiracy, and gene therapy. See, what happens is when you have a gene and then there is a targeting involving gene amplification, okay? So we have a gene amplification technique to treat a disease. So what are we doing here? See, what happens here is our kids get confused between these two options, gene therapy and molecular diagnostics. Okay, how? See here, gene targeting involving gene amplification in attempt and in attempt. Okay, is attempted in individual tissue to treat a disease. What is the technique called as is the question asked. Okay, so you have to cleanly observe the question. So then when why do they ask gene amplification? And they, why do we talk about gene amplification in gene therapy? No, right? Where do we see gene amplification? I just gave you an example. For example, okay, for example, we have a patient suffering from HIV. Okay, HIV. Now, what happens to this patient's kids? He has a cell, our human cells. Basically, we have T4 helper cells. Helper cells of our, there are helper cells in our immune system. So basically, those cells are targeted by our HIV viruses. Now, what happens to these cells is our HIV, not our HIV virus comes and it injects its DNA through a capsid and its RNA. Basically, its genetic material is RNA, right? So the RNA enters into the cell. The RNA undergoes reverse transcription. So the viral RNA is converted into a viral DNA, which is single stranded through reverse transcription. Okay, reverse transcription. Once it performs a reverse transcription, we have a single stranded DNA, and the single stranded DNA gets incorporated into the human cell. Okay, somewhere here. So this is what has happened in the patient who is suffering from HIV. Let's say that. Now, how do we detect them? We have two sets of individuals. One who we suspect that this person might have HIV, and the person who we think that he is healthy. So let's say we take the healthy person's gene, similarly, like how I taught in the last class, last process. This is the healthy gene, this is the HIV gene, and HIV infected cell. So in HIV infected cell, there is viral DNA. A part of a DNA is viral DNA, and rest of the gene is human gene. Now, what will happen? We will amplify this. When we amplify this viral gene gets becomes more and more. Even other genes also becomes more and more along with it. So now what we do, we take this normal genes and then we radioactive label it. Okay, when we radioactive label it, now it will become a probe DNA. So this probe is also called as hybridization probe. So this probe, what we will do, it will complement to the DNA of humans, but it will not complement to the DNA of virus. So now what will happen? All the human DNA will get complemented. Okay, the viral DNA will get no complementation because we don't have the complement for it. So when we see a parts or fragments of DNA that is not complemented, that means it is having a viral DNA. So that shows that this person is suffering from so and so disease. Understood? That is called as molecular diagnostics. So your answer is A. Understood? It's a PCR technique. Clear? Let's go next. But it's not gene therapy. Gene therapy is you detect the disease and then you treat a disease. It does not include gene amplification. Okay, you just detect the disease. Okay, so that is called as gene therapy. Next one. Here is our next question. Nowadays, it is possible to detect the mutated gene causing cancer by allowing the radioactive probe to hybridize its complementary DNA in clone of cells, following by its detection using autoradiography. Because, see, just now we discussed, the question is coming based on that, and it was asked in 2021. So let's see what are the options. Mutated genes does not appear on a photographic film as the probe has no complementarity to it or with it. Our second option is mutated genes does not appear on photographic plate or film as the probe has complementarity with it. Next one, mutated gene partially appears on a photographic film. Mutated gene completely and clearly appears on a photographic film. It should be super easy for you to answer this. Okay, so let's see if you get this. I will read the question once again. Nowadays, it is possible to detect a mutated gene causing cancer by allowing the radioactive probe to hybridize its complementary DNA in clone of cells, following the detection using autoradiography because. So this detection happens using autoradiography because, and you have the options. So let's see. Let's see if you guys are answering or if you have an answer. Oh, D. Okay. Is there any answers? I understand that you guys are sitting in groups in your classroom, and I would encourage it if you discuss among yourself and tell out the answers, okay? And compete among yourself, fight among yourself to get the right answer, okay? Healthy fighting. Anyways, so let's see the answer for this. If you look at this question, it is similar to that of molecular diagnostics in the detection of a disease. What did we see? That just in two questions also we saw the similar type of a situation, and it's the same technique. So what are we doing? How is it possible to detect a mutated gene or a viral gene? You take, Baba. So last in HIV also we are taking about viral gene. In prior case we are talking about some other thing, right? So similarly, let us talk about this, which is respect to cancer. So there is a gene mutation in a cell, so that is leading to a cancer. So now what we are doing, we want to detect if the cell has a cancer by radioactively labeling a probe. Now the probe will hybridize to its complement, correct, right? So the DNA in the clone of cells following by its detection using autoradiography. How is it possible for us to detect whether it is a cancer cell or a not cancer cell is a question. That is possible because if this is one healthy cell, okay, this is healthy cell and this is cancer cell, okay, this is cancer cell. Now what happens? Healthy cell has healthy gene. No, cancer cell has a gene. Among that, a part of a gene is mutated. Now how do we detect it? We take a healthy gene, we do radioactive labeling, same thing, same technique, and we take a cancer cell and we amplify the gene. Amplify means we increase its numbers so that we have enough volume or enough number of this cancer part or mutated gene part. Now what do we do when we take this radioactive labeled probe? This is called as probe, the healthy DNA, and we hybridize with our cancer cells or cancer gene. What will happen? All the healthy parts will get hybridized, okay, because we have complement to it. A healthy cell will not have cancer gene, right? So only unhealthy cell have cancer gene. So our healthy cells, whatever the probe that we are using, will only complement the healthy parts of the gene. The mutated parts of the gene remains unhybridized. Okay, it will remain unhybridized. So when it remains unhybridized, that means what is hybridization? I am a radioactive labeled probe. Now I will go and attach to my complement. So when I go attach to my complement, when I shine some light of UV radiation or UV light, when I shine it on me, now both of us will glow. Okay, both of us will glow. Whichever I go and hybridize, attach, so that will glow. Whichever do not get complemented or attached, that will not glow. That is called as non-hybridizing. So that happens why? Because mutated genes does not appear on the photographic film. Why it will not appear? Because it does not have the hybridized probe. Okay, because as the probe has no complementarity with it. The option is A. Okay. Is it clear? Give me a reaction. Let me see if you guys are listening. Yeah, wait, let me see. Yes, did you understand? So that is the last question. Oh, for last question you answered. Okay. I get you. Now, did you guys understand this concept? Good. Good. Okay, let's go for the next question. Okay. What do we have here? So with regard to insulin, choose the correct options. C-peptide is not, okay, C-peptide is not present in mature insulin. The insulin produced by DNA technology has C-peptide. The proinsulin has C-peptide. A peptide and B peptide of insulin are interconnected by disulfide bridges. Choose the correct options or answers from the given options below. Our A option says A, C, D only. Our B option says A and D only. Our C option says B and D only. And our D option says B and C only. Okay. The question is about insulin. Okay. Which of the following with regard to insulin is correct? Okay. So C peptide is not present in mature insulin. The insulin produced by the DNA technology has C peptide. The pro-insulin has C peptide. A peptide and B peptide of the insulin are interconnected by the disulfide bridges. Okay. So without wasting time, let's go for the answer. So we have to find the correct options. First option, let us see. C peptide is not present in mature insulin, correct, right? That is why when Eli Lilly tried to design our or genetically engineer or produce our human insulin, they faced the problem. Why? Because in the beginning, when they took the whole insulin gene and they put it into E. coli, it produced not mature insulin, it produced pro-insulin. What is pro-insulin? Our pro-insulin has 3 peptide chains. Okay. What are these peptide chains? I hope you know what is a peptide chain. Okay. A peptide, B peptide, C peptide chain. If people are wondering what is he talking about peptide, see what is a peptide chain? Proteins are made up of peptide chains. Peptide chains are made up of polymer of amino acids. You know what are amino acids, right? So amino acids are basic building blocks of your protein. So the chain of amino acids will lead to one type of peptide chain. The combination of peptide chain will lead to proteins, or the modification of peptide chain will lead to functional proteins. But here, when you see the the pro-insulin will have 3 chains, that is A, B, and C peptide chains. So since the C peptide chain should not be present in the pro-insulin, mature insulin, only A and B peptide should be there in the mature insulin. So C peptide is not present in mature insulin is correct answer. Okay. So what happens? The C peptide gets removed in our human body where pro-insulin is converted into mature insulin by removing the C peptide. Understood? Only A and B will be there. So that is why first option is correct. The insulin produced by the DNA technology has C peptide. No. Before it was. In the beginning, they experimented, they got pro-insulin. So that is why what this company did, they separated this A peptide producing gene, B peptide producing gene, okay, A and B peptides producing gene. They separated this gene and they inserted into 2 different E. coli. This is one E. coli, this is another E. coli. So now this E. coli produced A peptide and this produced B peptide. Finally, they combined it to produce mature insulin. So the recombinant insulin do not have C peptide. So this option is wrong. And the third option, the pro-insulin has C peptide, correct? A peptide and B peptide of insulin are interconnected by disulfide bridges. Yes. When these peptide chains are formed, the A peptide and B peptide, they are interconnected by disulfide bridges to hold the chains together. So that is what creates them a mature insulin. So our option A, our option C, and our option D is correct. So A, C, and D. So our option A is the correct option for our question number 7. Next one. For effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which one of the following molecular diagnostics techniques is very useful for early detection? Okay, our options are ELISA technique, hybridization technique, Western blotting technique, Southern blotting technique. Okay, so we have these. Let's see what we have. So the given question asks about early effective treatment. For effective treatment of a disease, early diagnostics have to be done. See, you can go for permutation and combination in this odd one out, or you can call it as, so if you don't know the answer, you can go for odd one out. Southern blotting and Western blotting techniques are the blotting techniques where the fragments of DNA are transferred or RNA are transferred onto a nitrocellulose membrane. So when we transfer, see, when we do the agarose gel electrophoresis, right, the bands get separated in the gel and these bands are transferred onto a nitrocellulose membrane, correct, right? So when we do that, that is called a Southern blotting. This is for DNA. So if we do it for RNA, it is called as northern blotting. If we do it for protein, it is called as western blotting. So here only the transfer of pro or DNA fragments take place onto the membrane. That's all. So here it does not help in our early detection of a disease. Hybridization techniques. This is a technique where we try to see which complements with what match, finding a match. That's all. This also will not help us in diagnostics. Among these three things, which one helps us in diagnostics? ELISA. What is ELISA? Enzyme-linked immunosorbent assay, which is an immunological assay. Okay. For example, this assay is used for the detection of HIV virus. Okay. So our option A is correct with respect to this question. Okay. Let's go for the next question. ADA deficiency results into what? Digestive disorder, Addison's disorder, dysfunction of immune system, Parkinson's disorder. Okay. ADA. What does ADA lead to? Can you answer this question? This should be easy, right? Yes. Students, what does ADA deficiency lead to? Okay. So let's see what's the answer. ADA deficiency, adenosine deaminase deficiency. So ADA is an enzyme that is very much useful for your functioning, okay, healthy functioning of your immune system. Basically, ADA deficiency will affect our cell-mediated immunity. We have two types of immunity. One is cell-mediated immunity, other one is humoral immunity. So what happens is the cell-mediated immunity is very, very important for specifically targeting a pathogen and deactivating it effectively. It's like targeted immune response, you can tell. So whenever there is no ADA, what will happen is that will lead to a severe problem in reducing the immunological function. So when severely the immunological function is disrupted, a person will start showing syndrome. Okay. So what is a syndrome? They will show multi-symptomatic condition. So if a person shows malaria symptoms or dengue symptoms, they are having a disease. If a person is showing multiple symptoms of multiple diseases, that is called as a syndrome. So what happens to this? They actually start showing severe, okay, severe immunodeficiency syndrome, okay, or combined immunodeficiency syndrome. So that is what ADA leads to, okay, dysfunction of immune system. Okay, ADA deficiency lead to dysfunction of immune system. So that is a third option is the option for your question. It's easy. Okay, next we have another question. BT cotton variety that was developed by an introduction of this toxin gene of Bacillus thuringiensis is resistant to what? Very easy again. Plant nematodes, okay, then insect predators, insect pests, fungal diseases. Okay. So the question was asked in NEET 2020. So read the question again. BT cotton variety that was developed, okay, and introduced and by the introduction of the toxin gene of Bacillus thuringiensis is resistant to plant nematodes, insect predators, insect pests, and then we have fungal diseases. So when we talk about this question, we know that the BT cotton has a cry protein or cry genes, which is actually a pest-resistant gene. We have studied about this pest-resistant gene. So please do not combine these two. Our answer is C only. So BT cotton, the cry protein or the cry genes is always pest resistance. So what kind of pest are we talking about? Insect pest, bollworm, corn borers, right? I gave a list of these pests that are killed by using our Bacillus thuringiensis gene. Correct, right? Why don't we use plant nematodes? We don't apply to the plant nematodes. We saw in case of Melodygine incognitia that plant nematodes, we use a different technique called as RNAi technology, that is RNA interference technology. But when it comes to Bacillus thuringiensis, we have used it for the neutralization of plant, insect pests. Okay, a cotton bollworm, okay, or it is corn borer, etc. Next one. Which one of the following is true for the golden rice? So you have the option. It has yellow grains because of a gene introduced from a primitive variety of rice. It is a vitamin A enriched, okay, with a gene from daffodil. It is pest resistant with a gene from Bacillus thuringiensis. It is drought tolerant, developed using agrobacterium vector. So when we talk about golden rice, it's very famous. Why? Because it is also called as a biofortified rice. Okay. So biofortified or molecularly improved rice where it is enriched with vitamin A. So it's a GMO, genetically modified crop, where the gene from the daffodil is taken, okay, which is put into this rice, where this rice, instead of more carbs and fiber, it will produce more of vitamin A. So this rice was developed for to help the patients with night blindness. Okay, so that is why this golden rice is very famous for having vitamin A. And where it is coming from? Daffodil. That is a correct option, correct statement. So our next question states, in RNAi, okay, sorry, in RNAi, the genes are silenced using. So it's a 19 question. I talked about it again. RNAi, the genes are silenced using. Our options are double stranded RNA, single stranded RNA, single stranded DNA, single stranded RNA, and double stranded DNA. So these are our options. So when we talk about RNAi or RNA interference technology, what happens is it's called as gene silencing. What do? We take transposons, which are jumping genes, which can create several different permutations and combinations of RNAs randomly. So we take this kind of an element and we put it into a plant. Now, what will happen? Let's say an insect like a nematode comes and then it infects and it has its RNA. Now our transposons are producing its complementations. So once it matches, it will create double-stranded RNAs. So when it creates double-stranded RNA, double-stranded RNAs are simply useless. So that's how we silence a gene. How do we do it? If we have an RNA that is causing a problem, we try to create the complementation of this RNA by using some of the genes. Example, transposon gene. Understood? So thereby creating a double-stranded RNA. So if you create a double-stranded RNA, the translation will not take place, protein production will not take place, thereby the gene expression is cancelled. So when cancellation of gene expression is called as what? Silencing of genes. Clear? Okay. You have any doubts? I just solve like 3-4 questions rapidly because you are responding. Yeah, is it clear? Did you understand? Give me a reaction fast. Yes, no? Good. Okay. Let's go for the next question. Okay. 1 second. Give me a second. I need to check this also. Okay, no problem here. Okay. We have our next question. Which one of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Retroviral viruses, TI plasmids. This is phage. Okay. This is lambda phage. Okay, this is called as lambda phage vectors, and this is PBR 322. So the question is, which one of the following is commonly used as a vector for introducing a DNA fragment into the human lymphocyte? So I spoke about it, right? Lab-grown retroviruses are used. So why only lab-grown retroviruses are used? Why not the normal retroviral, viruses are used? See, when you use a lab-grown retrovirus, we make sure that this retrovirus do not have the capacity to divide. It can't divide. This is cancelled. All it can do is take our gene of interest, insert into our host cell, which is our lymphocyte. So the type of a vector we are using for the introduction of our ADA gene into our lymphocyte was retrovectors, and it will be retrovectors. Okay. So our option is option A. TI plasmid vectors, phage vectors are used for bacteria. Okay, bacteria, if it is a host, we use TI plasmid or phage vectors. And you know that PBR 322 is a plasmid vector. So you know who has plasmid. Bacteria have plasmid. Understood? Clear? Next one. In India, the organization responsible for assessing the safety of introducing genetically modified organisms for public use is which of the organism that is responsible for assessing the safety? Is it okay or not okay? Who does that? Which organization does that? So our option is Indian Council of Medical Research, ICMR, then Indian Council for Scientific and Industrial Research, that is CSIR, Research Committee on Genetic Manipulation, RCGM, Genetic Engineering Appraisal Committee. So, the organization in our country that is responsible for assessing the safety of introducing GMOs to the public use is, remember this, note it down, Genetic Engineering Appraisal Committee. So, this is the committee in our country that guards and takes responsibility of assessing the safety if it is okay for the public introduction of any GMOs. Clear? So take this down. Now, a new variety of rice was patented by a foreign company. Okay, through such varieties have been present in India, though such varieties have been present in India for a long time. This is related to this case is related to what? CO667. Okay, then Sharbati Sonora. Then we have Lerma Roja, and then we have Basmati. This is very obvious for us. When we talk about a particular rice variety that was patented by a foreign company, obviously we go for Basmati. So Basmati rice had a problem. What happened? A Texas company called as Rice Tech, they took our Basmati rice varieties and they developed a new variety called as semi-dwarf varieties of Basmati. And they said that we are the novel inventors or discoverers of this variety. But for centuries, we are using 27 varieties of basmati rice in our country. So that is when India threatened USA against legal action. So USA was very powerful back then. They were like, foreign policy. Foreign policy was, quite. It became very, very tight because of this case. But India did not back down. They told USA that we are going to take the case for World Trade Center. Okay, and they filed a case against this practice, and finally the case was in our favor, and the company lost a lot of hold on this particular research. Understood? So next one. Use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called. Our options are bio-infringement, biopiracy, biodegradation, bioexploitation. Okay. So now, use of bioresources by multinational companies and organizations without authorization from the concerned country. So similarly, like I told you, right, indigenous countries or ancient cultural countries, culturally rich countries, or if you see any second world countries, we have more association with our nature than industrial countries. So we have a lot of traditional knowledge. When this traditional knowledge is exploited for their own benefit and they make money out of it, so that is called as biopiracy without permission if they do it, like turmeric, neem, okay, charcoal, whatever we have been using for centuries, they think that they have discovered the medical use and they try to make money out of it, which is not permitted, right? So that is not right to exploit nature for their own thing. So that is called as biopiracy. Okay. So this one is easy. We have solved it. Okay. This is a very interesting question. Now, what happens? The crops engineered by glycophosphate are resistant or tolerant to, okay, bacteria, insects, herbicides, and fungi. Now, what will happen? There is a genetically engineered crops, okay, which are, most of the crops are genetically engineered now for a particular purpose that is called as glycophosphate, okay, resistant. Now, what will happen? There are two plants you take. One crop, okay, one crop with respect to, let's say, one crop with respect to, this is a normal crop, okay. Other crop is a recombinant crop. And this recombinant crop has glycophosphate modification. And this normal crop do not have glycophosphate modification. Now, what will happen is, whenever a herbicide is sprayed, you know that, right? In our farm, herbs, Okay, herbicide will grow, which is not okay. So whenever a herbicide is sprayed on this, now what will happen is, usually the crop which is normal, for it a glycophosphate pathway is interrupted. When a glycophosphate pathway is interrupted, now usually in algae and in higher plants, okay, so this glycophosphate is very very important, okay, for plant growth and development. So when that is completely disrupted, this crop will die. This crop will actually die. So basically in broadleaf plant, this problem will happen. So when we have a recombinant crop where the glycophosphate pathway is not interrupted, so flawlessly this glycophosphate will be synthesized. So in this, it will not get affected. Since here, when the herbicide is sprayed, glycophosphate gets destroyed, okay, or disrupted, this crop will die. Here it will not die. So that is why this particular genetic engineering is used, okay, for us herbicides, okay. Against herbicides, if the plant has to be resistant, we use glycophosphate engineering. All right, students, we come to the end of the session. Okay, if you have any doubts, you can ask me. Thank you very much. Have a nice weekend. Okay, Basmati, biopiracy. Sir, see my answers. Yeah. Sorry. Veera Venkatraju, Malayalam. I could not see because we were running out of time. Okay, good. Anyway, you have given the right answers. Okay. So students, have a nice day. See you tomorrow. Okay. Take care.